Hey guys, welcome to Liver and Glucose Homeostasis. I'm your host, and we're here with an AI, Grok. It's going to be explaining the topics for us. I'm unfamiliar with the topics myself, this is the whole point. But I'm going to be asking the questions, so hopefully we can learn something throughout this process. Take everything that AI says with a pinch of salt, and this is part one of two. It's going to be 30 minutes long. Okay, let's start, Grok. Give us an introduction on the liver and glucose homeostasis.
All right, let's dive into the liver and glucose homeostasis. The liver is a key organ that regulates blood sugar levels by storing excess glucose as glycogen or releasing glucose into the blood when needed, maintaining energy balance. Glucose homeostasis is the process of keeping blood glucose levels stable, involving hormones like insulin and glucagon, with the liver acting as the central hub for glucose production, storage, and release. Ready to start with a specific aspect, like how the liver handles glucose or the hormones involved?
I want to ask you about different liver functions. I mean, we know it stores glycogen, and we know it breaks down glycogen. We can synthesize glucose from non-carbohydrate precursors. We can deaminate amino acids and convert them to convert ammonia to urea. Explain this. I'm not very sure about this last one. And what other things does it do?
The liver has several key functions in glucose homeostasis and beyond. It stores glucose as glycogenesis, breaks down glycogen to release glucose, glycogenolysis, and synthesizes glucose from non-carbohydrate precursors like lactate, amino acids, or glycerol through gluconeogenesis. It also deaminates amino acids, removing their nitrogen groups to form ammonia, which it converts to urea for safe excretion via the urea cycle, detoxifying the blood. Beyond glucose, the liver metabolizes lipids, produces bile for fat digestion, detoxifies drugs and toxins, and synthesizes proteins like albumin and clotting factors. Want to zoom in on one of these, like the urea cycle or gluconeogenesis?
Well, tell me about the synthesis of fatty acids from glucose a bit, and the synthesis of ketone bodies too.
The liver synthesizes fatty acids from excess glucose through lipogenesis, where glucose is converted to acetyl-CoA in the cytoplasm, then built into fatty acids by enzymes like fatty acid synthase, storing energy as fat. When glucose is scarce, the liver produces ketone bodies acetoacetate, beta-hydroxybutyrate, and acetone via ketogenesis in mitochondria, breaking down fatty acids to provide an alternative energy source for tissues like the brain during fasting or starvation. These processes help the liver manage energy balance, shifting between storing glucose as fat or generating ketones when glucose levels drop. Want more on how these processes are regulated or their role in glucose homeostasis?
Let's think about so we need ATP, so we need the glucose from the glycogen, we need the fatty acids, we need the amino acids. So we get these from meals, and we also need to store them because maybe we need them more at other points. So explain to us how we're able to store fuels when they are abundant.
When fuels like glucose, fatty acids, and amino acids are abundant from meals, the liver stores them to manage energy needs. Glucose is converted to glycogen via glycogenesis for short-term storage, or to fatty acids through lipogenesis for long-term storage in adipose tissue, while excess amino acids are deaminated for gluconeogenesis or converted to fat. Insulin, released after eating, promotes these storage processes by activating enzymes and glucose uptake, ensuring energy reserves for when food is scarce. Want more on insulin's role or how these stores are used later?
Okay. So we need also to release these fuels in a controlled way. So how does that occur? Is it through these hormones?
Yeah, the liver releases stored fuels like glucose, fatty acids, and ketone bodies in a controlled way, mainly driven by hormones like glucagon and epinephrine. When blood glucose is low, glucagon triggers glycogenesis to break down glycogen into glucose and gluconeogenesis to make glucose from amino acids or glycerol, while epinephrine boosts these during stress. Low insulin also signals fat breakdown, releasing fatty acids and triggering ketonegenesis for energy. These hormones ensure fuels are released to meet energy demands, like during fasting or exercise. Want to dive deeper into a specific hormone or
So
process?
after a meal, we have absorptive events happening. We have liver and adipose tissue taking up materials, and we have post-absorptive events too. So after the meal, between meals, we have exporting from the liver and adipose tissues.
After a meal, during the absorptive phase, the liver and adipose tissue take up glucose and lipids, driven by insulin, which promotes glycogenesis and lipogenesis to store energy as glycogen and fat. In the post-absorptive phase, between meals, the liver releases glucose via glycogenesis and gluconeogenesis, and adipose tissue breaks down fats to release fatty acids, both triggered by glucagon and low insulin to maintain blood sugar and energy supply. These processes ensure energy homeostasis by storing fuels when abundant and releasing them when needed. Want to focus on specific triggers or metabolic pathways?
Tell me about energy stores now. Specifically, what I want to understand is how do amino acids relate to oxaloacetate, acetyl-CoA, and pyruvate? And how do fatty acids relate with acetyl-CoA and fatty acids?
Amino acids can be broken down in the liver to produce pyruvate or oxaloacetate, which feed into gluconeogenesis to make glucose. Or they can form acetyl-CoA, which enters the Krebs cycle for energy, or is used for ketonegenesis. This depends on the amino acid structure, like glucogenic ones making pyruvate or oxaloacetate, or ketogenic ones yielding acetyl-CoA. Fatty acids are broken down via beta oxidation in mitochondria to produce acetyl-CoA, which fuels the Krebs cycle for ATP production, or, during fasting, is converted to ketone bodies for energy. Fatty acids themselves are not synthesized from other fatty acids, but stored as triglycerides. These processes connect dietary amino acids and fats to energy production and glucose homeostasis in the liver.
I don't understand how they're related. Can you very simply explain it step by step? Just not too many words, though.
Okay, step by step, super simple. Amino acids are broken down in the liver. Some turn into pyruvate or oxaloacetate, which make glucose via gluconeogenesis, while others form acetyl-CoA for energy in the Krebs cycle or ketone bodies. Fatty acids, through beta oxidation in mitochondria, become acetyl-CoA, which fuels the Krebs cycle for ATP or makes ketone bodies during fasting. These processes link amino acids and fatty acids to energy and glucose production in the liver. Want to focus on one part, like gluconeogenesis?
What even is acetyl-CoA oxaloacetate and the citric acid cycle? How do they relate? I know. So, can you? I don't get it. How do these relate?
Acetyl-CoA and oxaloacetate are key players in the citric acid Krebs cycle, which happens in the mitochondrial matrix to produce energy. Acetyl-CoA, formed from glucose, fatty acids, or amino acids, combines with oxaloacetate to start the cycle, forming citrate, which then undergoes reactions to generate ATP, NADH, and FADH2. Oxaloacetate is regenerated at the cycle's end, ready to combine with another acetyl-CoA, linking them as essential partners in energy production from various fuels. Want a deeper dive into the cycle steps?
And so amino acids become oxaloacetate and fatty acids become acetyl-CoA.
Yeah, some amino acids are broken down in the liver to form oxaloacetate or pyruvate, which can feed into gluconeogenesis or the Krebs cycle, while fatty acids are broken down via beta oxidation to form acetyl-CoA, which enters the Krebs cycle or makes ketone bodies. Both connect to energy production, with oxaloacetate and acetyl-CoA combining in the Krebs cycle to generate ATP. Not all amino acids become oxaloacetate. Some directly form acetyl-CoA depending on their structure. Want more on specific amino acids or the Krebs cycle?
So how do amino acids and fatty acids become pyruvate and acetyl-CoA and oxaloacetate? What is the process?
Amino acids are broken down in the liver through deamination, where their nitrogen is removed, and their carbon skeletons are converted into pyruvate for glucogenic amino acids like alanine or oxaloacetate like aspartate, or directly into acetyl-CoA for ketogenic amino acids like leucine. Fatty acids undergo beta oxidation in mitochondria, where they're chopped into 2 carbon units, forming acetyl-CoA, which can enter the Krebs cycle or make ketone bodies. These processes, deamination for amino acids and beta oxidation for fatty acids, channel dietary fuels into energy production or glucose synthesis via intermediates like pyruvate, oxaloacetate, or acetyl-CoA. Want more on specific pathways or examples?
Yeah, I want to go a bit deeper. So let's start with amino acids and then we'll talk about what happens with fatty acids so we start with an amino acid. How does it end up being pyruvate, acetyl-CoA, or oxaloacetate?
Amino acids are broken down in the liver via deamination, where enzymes remove their nitrogen group, forming ammonia, later converted to urea, and their carbon skeletons are transformed into pyruvate, e.g. from alanine via transamination, oxaloacetate, e.g. from aspartate, or acetyl-CoA, e.g. from leucine for ketogenic amino acids. These intermediates then feed into gluconeogenesis, pyruvate and oxaloacetate to make glucose, or the Krebs cycle, acetyl-CoA for ATP production, depending on the amino acid structure and the body's energy needs. The process starts in the cytosol for transamination and continues in mitochondria for some conversions, tying into glucose homeostasis or energy supply. Want me to break down fatty acid metabolism next, or focus on a specific amino acid?
Let's talk about glucose as an energy substrate then. So we've got diet, we've got gluconeogenesis, we've got liver glycogen breakdown, all these things. And then this is gonna go to the blood, and some of it is gonna go to the body and to the brain.
Glucose, a key energy substrate, comes from dietary carbohydrates, liver glycogen breakdown, glycogenolysis, or gluconeogenesis using precursors like lactate, amino acids, or glycerol. The liver releases glucose into the blood, supplying energy to tissues like muscles and the brain, which relies heavily on glucose for ATP production via glycolysis and the Krebs cycle. Hormones like insulin and glucagon regulate this release to maintain stable blood glucose levels for body-wide energy needs. Want to dive into how specific tissues use glucose or the hormonal controls?
Yeah, let's talk a bit about the brain. So the brain is very vulnerable. Well, it's the most vulnerable to hypoglycemia. It's a bit picky with what it accepts as to pass the blood-brain barrier, from what I understand. Correct me if I'm wrong. So the cerebral cells, they mainly get their energy from aerobic metabolism, and they don't really store glucose in much amounts, and they don't synthesize glucose, and I wonder why. And they also don't metabolize substrates other than glucose or ketone bodies, and I wonder why about that too.
Brain cells, especially neurons, rely on aerobic metabolism for energy, using glucose from the blood via glycolysis and the Krebs cycle, because they lack significant glycogen stores and can't synthesize glucose, as they don't have gluconeogenesis enzymes. prioritizing rapid ATP production for high energy demands. The blood-brain barrier limits substrate access, allowing mainly glucose and, during starvation, ketone bodies, produced by the liver from fatty acids to cross, as other substrates like fatty acids are too large or lack specific transporters. This specificity ensures efficient, controlled energy supply, but makes the brain vulnerable to hypoglycemia if glucose or ketone body levels drop. Want to dive into how ketone bodies support the brain or hypoglycemia effects?
Tell me about why they can't extract much glucose for their needs from extracellular fluids at low concentrations. Yeah, is this to do with hormones not facilitating glucose in the brain?
Brain cells, particularly neurons, struggle to extract glucose from extracellular fluid at low concentrations because their glucose transporters, like GLUT3, rely on a concentration gradient, and low blood glucose, hypoglycemia, reduces this gradient, limiting uptake. Hormones like insulin don't directly facilitate glucose transport into brain cells, as neuronal GLUT3 is insulin independent, but insulin influences overall blood glucose levels by promoting uptake in other tissues, indirectly affecting brain availability. During hypoglycemia, the brain's inability to store or synthesize glucose, combined with its high metabolic demand, makes it dependent on constant blood glucose supply or ketone bodies as backup. Want to dive into glucose transporters or hormonal regulation further?
I want to ask you about mechanisms controlling blood glucose. So we've talked about gluconeogenesis, we've talked about...
Blood glucose?
Wait, we've talked about glycogenolysis. How do catecholamines play a role? How does cortisol play a role? How does growth hormone play a role?
Catecholamines, like epinephrine, trigger glycogenolysis by activating enzymes to break down liver glycogen into glucose, rapidly raising blood sugar during stress. Cortisol promotes gluconeogenesis, using amino acids and other precursors to produce glucose, while also reducing peripheral glucose uptake, aiding long-term blood sugar maintenance. Growth hormone increases blood glucose by stimulating glycogenolysis and gluconeogenesis in the liver and decreasing glucose uptake in tissues, contributing to glucose homeostasis during growth or stress. Want to focus on one hormone's mechanism or their combined effects?
Explain to me again about these, what they do exactly.
Catecholamines, like epinephrine, bind to liver cell receptors, activating adenase cyclase to increase CAMP, which triggers glycosylsclosism by phosphorating enzymes that break glycogen into glucose for quick blood sugar spikes. Cortisol, a glucocorticoid, upregulates gluconeogenesis enzymes in the liver, converting amino acids and glycerol into glucose, and reduces glucose uptake in muscles and fat to sustain blood sugar over time. Growth hormone stimulates liver glycogenolysis and gluconeogenesis via signaling pathways like JKST, while also promoting insulin resistance in tissues, elevating blood glucose for energy demands during growth or stress. Want a deeper dive into one of these pathways?
What's going on during sleep? So, is there a dependence on de novo glucose synthesis? Can you explain this? Basically, gluconeogenesis kicks in sleep.
Yeah. During sleep, gluconeogenesis ramps up in the liver to produce glucose from non-carbohydrate sources like amino acids and glycerol, maintaining blood sugar levels since you're not eating. This process, driven by glucagon and cortisol, ensures a steady glucose supply for the brain and other tissues overnight. Glycogenolysis also contributes initially, but gluconeogenesis becomes key during prolonged fasting states like sleep. Want more on how hormones regulate this or its impact on specific tissues?
Did others Homo have this? Homo Neanderthal and all these lots, Erectus? Did they have this skill with sleep?
Gluconeogenesis during sleep, crucial for maintaining blood glucose levels, is a fundamental metabolic process likely present in all Homo species, including Neanderthals and Homo erectus, as their brains and bodies required steady energy, especially for high-energy tissues like the brain. While we lack direct evidence from fossils, their shared ancestry with Homo sapiens and similar physiological demands suggests they relied on gluconeogenesis, driven by hormones like glucagon and cortisol, during fasting states like sleep. Differences in diet or activity might have tweaked its regulation, but the core mechanism was probably conserved across hominins. Want to dive into how their diets might have influenced this?
I want to ask you about hormonal control. So we've got the main men or women, insulin, glucagon. So insulin will take the glucose to the liver, glucagon will break it down. Let's talk a bit more about this. So how exactly does insulin and glucagon act? Because besides the vague, description of I just gave.
Insulin, released by pancreatic beta cells when blood glucose is high, binds to receptors on liver cells, activating pathways that promote glucose uptake, glycogenesis, storing glucose as glycogen, and lipogenesis, while inhibiting gluconeogenesis and glycogenolysis. Glucagon, released by pancreatic alpha cells when glucose is low, binds to liver receptors, stimulating glycogenolysis to break down glycogen into glucose and gluconeogenesis to produce glucose from non-carbohydrate sources, releasing it into the blood. These hormones act oppositely to tightly regulate blood glucose, ensuring energy availability or storage based on the body's needs. Want more on their signaling pathways?
Yes, let's break them step by step. So you mentioned pancreatic alpha cells. Let's start with glucagon.
Glucagon, released by pancreatic alpha cells when blood glucose is low, binds to G protein-coupled receptors on liver cells, activating adenase cyclase to produce CMA, which triggers protein kinase A. This kinase phosphorates enzymes like glycogen phosphorylase, driving glycogenolysis to release glucose from glycogen, and upregulates gluconeogenesis enzymes to make glucose from lactate, amino acids, or glycerol. The glucose is then released into the bloodstream to raise blood sugar levels, especially during fasting or sleep. Want to move to insulin's mechanism or dig deeper into glucagon's pathways?
Yeah, how exactly does it do what it does? Same with insulin.
Glucagon binds to liver cell receptors, activating CMA and protein kinase A, which phosphorylates enzymes to trigger glycogenolysis, breaking glycogen into glucose, and gluconeogenesis, making glucose from non-carbs, raising blood sugar. Insulin, released when glucose is high, binds to liver and muscle cell receptors.
So with glucagon, so it binds to liver receptors, cell receptors, and you said something about CMA and it activates protein kinase A, and what exactly does this do?
Glucagon binds to G protein-coupled receptors on liver cells, activating adenase cyclase, which produces CMA, a signaling molecule that activates protein kinase A, PKA. PKA phosphorylates key enzymes, like glycogen phosphorylase, triggering glycogenolysis to break down glycogen into glucose, and upregulates gluconeogenesis enzymes to produce glucose from non-carbohydrate sources. This raises blood glucose levels to meet energy demands during low sugar states like fasting. Want to dive into insulin's pathway next, or focus on something specific?
Yeah, let's do the insulin pathway now.
Insulin, released by pancreatic beta cells when blood glucose is high, binds to tyrosine kinase receptors on liver and muscle cells, activating the PI3K-ACT pathway, which promotes glucose uptake via GLUT4 transporters and stimulates glycogenesis enzymes to store glucose as glycogen. It also inhibits glycogenolysis and gluconeogenesis by suppressing relevant enzymes, ensuring glucose is stored or used rather than produced. This lowers blood sugar and supports energy storage for future needs. Want to dive deeper into a specific step or compare it to glucagon?
So why? How come we name the alpha cells for glucagon and the beta cells for insulin? And tell me again what it does with GLUT4 and why specifically GLUT4?
Alpha cells in the pancreas produce glucagon and beta cells produce insulin, named historically based on their order of discovery and distinct roles in glucose regulation, with alpha cells responding to low glucose and beta to high glucose. Insulin binds to receptors, activating the PI3K-ACT pathway, which moves GLUT4, glucose transporter type 4, to cell membranes and muscle and fat cells, allowing glucose uptake to lower blood sugar. GLUT4 is specific because it's insulin responsive, unlike other glucose transporters, and is critical for rapid glucose entry into energy-using tissues. Want more on the naming history or GLUT4's mechanism?
Yes.
Cool. So you want to dive deeper into the naming history of alpha and beta cells or the specifics of GLUT4's mechanism?
Both.
Alpha and beta cells got their names from early histological studies, where they were labeled based on their pancreatic islet positions and staining patterns. Not much more to it. For GLUT4, insulin triggers its movement from inside the cell to the membrane via vesicles, letting glucose flood in to be used or stored, specific to insulin-sensitive tissues like muscle and fat. Pick a focus or something new to keep it rolling.
Okay. Let's talk a bit about the entry of glucose into cells from the blood. So how do polar molecules like glucose enter the cells across a lipid membrane?
Polar molecules like glucose can't cross the lipid membrane easily due to its hydrophobic nature, so they enter cells via facilitated diffusion through specific glucose transporters, primarily GLUT proteins. In insulin-sensitive tissues like muscle and fat, insulin triggers GLUT4 to move to the cell membrane, allowing glucose to enter from the blood for energy or storage. Other tissues, like the liver, use GLUT2, which is always present and facilitates glucose uptake based on blood glucose concentration gradients. Want to dive deeper into GLUT transporters or their regulation?
So you're saying because... So why does something that's polar not like a lipid membrane? And polar basically means there's large electronegativity?
Polar molecules like glucose have uneven charge distribution, making them hydrophilic and unable to pass through the hydrophobic, nonpolar lipid bilayer of cell membranes without help from transporters like GLUT4 or GLUT2. The lipid membranes...
Why does an uneven charge mean you cannot pass through?
Polar molecules like glucose have uneven charge distribution due to electronegative atoms, like oxygen and glucose's hydroxide groups, creating partial positive and negative regions that don't mix well with the nonpolar, hydrophobic lipid bilayer of cell membranes. This mishmash prevents them from passing through without transporters like GLUT4, which provide a specific channel for glucose to cross into cells. The uneven charge makes polar molecules sticky with water, not the oily membrane, needing facilitated diffusion to enter. Want more on how transporters work or why polarity matters?
That's all good. I just want you to end with a sentence now. This is part 1 out of 2. If someone would have taken away one thing from this episode, what would it be?
The liver maintains glucose homeostasis by storing glucose as glycogen when abundant and releasing it through glycogenolysis or gluconeogenesis when needed, regulated by insulin and glucagon. Thanks for tuning in. Catch you in part 2.
Okay, perfect. So, thank you for watching, guys. I hope you enjoy these 30-minute videos more than the longer ones. Let me know, and I'll see you in part 2. Bye-bye.